Okay, so far that's all for the pressure flow hypothesis. Okay, next I will explain about the another three type of hypotheses: electroosmosis, cytoplasmic swimming, and final one is peristaltic bath. Okay, so normally these three type of hypotheses is very cold topic that normally student will not learn it lah, because because too boring, or maybe not never come out yet, even in real exam never come out yet in both objective, structure, and even essay. So that is why that one of the reason nobody want to care about that lah. But however, because it is in the syllabus, so that you should know it. Okay, you should know too lah about these three type of hypothesis. Okay, so first is about the electroosmosis hypothesis. Okay, so when you see this term osmosis, so when you see the osmosis, this process will be using water one. It will including the water. And then if you see the electro, meaning that it will include electrical charge. So that so actually this one is one type of chemistry theory in electroosmosis lah, but in STPM syllabus we not learn that. So one of the reason that many people will be confused lah. So anyway, but if you don't know about the theory of electroosmosis, actually you still can remember it. Okay, so electroosmosis actually is the movement of ion in an electrical field through fixed porous with its electrical charge by carrying water and potassium ion. So there are three main components that were involved in electroosmosis. One of the main component is water molecule, and another one of course solute lah, because translocation we need to transport solute, right? So of course solute, and another one is potassium ion. Okay, so how it is become. So normally in your sieve tube and sieve plate and your film protein are normally in negatively charged, thus forming a fixed porous surface with an electrical charge. So normally in your sieve plate it is in negatively charged one. At your beginning process. Okay, and then when your flow occur through the negatively charged sieve plate, an ion will be repelled, but cation will be able to pass through. So you know that this is it at the initial process. Your sieve plate is negatively charged, right? I already mentioned your sieve plate is negatively charged because your protein is negatively charged, so that it will repel any anion molecule. We know that in electrical charge, anion and anion is repelled. Same electrical charge are repelled to each other. So only the cation molecule can only attract cation molecule. So what kind of cation molecule? So in this plant, potassium ion will play the role in cation molecule. Okay, so that your cation molecule, potassium ion, will be transported from the component cell into the sieve tube element. So when your mass flow occurs downward through the film, the repulsed ion will accumulate above the sieve tube, so that above sieve plate will become negative. And then sieve plate now will be fixed porous surface within an electrical field, such as is needed for electroosmosis to occur. Okay, so when there are differences occur across the sieve plate is reached, your proton or hydrogen ion from the wall of upper cell into each cytoplasm will lower the pH value, making the cytoplasm above the sieve tube become positively charged. Okay, so if you see this, so when you want to mass flow occur downward through film, your anion will be accumulate first at the sieve plate first, so that at your initial part, all your ion at here is totally negatively charged at initial process. Okay, so once that within a electrical field, so that it can produce a electrical field. Okay, and then when the critical potential difference is already across the shift plate is reached, hydrogen ion from the wall of the upper cell into each cytoplasm will lower the pH, and then making the above the shift tube element become positively charged. So hydrogen ion from the any part of the cell will start to diffuse into the shift tube element. Okay, so that your charge for the shift tube element at the above will become positively charged now. Okay, so this increased positively charge push other cation, mainly is potassium ion, by electrical pulsion through the shift plate from the upper to the lower cell, and therefore your electroosmosis will be occur. So your potassium ion then secreted on the another side of the shift plate, and then presence of potassium ion on the other side of the shift plate induce negatively charge on the other side of the shift plate. So it is actually like the macam play the charge lah, play the charge between the shift plate. So normally when the hydrogen ion is already accumulated at here, potassium ion will be accumulated at here, and then this positively charged ion will cause the potassium ion diffuse through the shift plate, and then when the potassium ion is already passed through, your solid molecule and the water molecule will automatically also will pass through, so that the electroosmosis will be occur. So this shared hydrated potassium ion will carry water molecule and dissolve solute across the shift tube. So with the energy cost on electroosmosis, flow of polar water molecule and dissolved solute through the shift pore to the adjacent shift tube element. So this is the overall process of the electroosmosis lah. So normally it play the charge between the shift tube element. You will see here it play the charge only. So initially at the beginning, your charge for the shift tube element is negatively charged. For the upper part is totally negative, and then when the hydrogen ion is already starting to pump into the shift tube element, potassium ion also will start to pump into the shift tube element, causing the upper part of the shift plate become positively charged. Okay, so when the upper part become positively charged, so you see this here. See here. So when the electric charge, so electroosmosis will be occur. So how it occur? Your positive ion on other side of the shift plate will induce negatively charge on the other side of the shift plate, creating a potassium difference across the shift plate, so that your potassium ion will starting to diffuse out, because it need to induce the charge at the lower part of the shift plate. So when the potassium ion is already passed through the shift plate, so that the water molecule and your solute will also pass through into it, so that when the potassium ion is already inside the pass through the shift plate, and then it will recycle back into the component cell, and then it will recycle again and again. So it is like based on the electrical charge lah. So yeah, okay. Another one is about cytoplasmic streaming hypothesis. Okay, so about the cytoplasmic streaming hypothesis, actually is the flow of the ion of the molecule that can be flow inside the cytoplasm. So normally it had been proposed that your solute can be carried from one end of a sieve element to other by streaming and then transfer the across sieve plate by active transport. So streaming is that your cytoplasm or plant cell is ordered to move around within the cell. So meaning that actually your organelles or your any nutrient or organelles inside the cell actually can be moved around freely one. So this process is called streaming. And then it found that when you see this diagram lah. Okay, so you see this diagram, any molecule inside the sieve tube element is freely moved one. It is very freely moved. And then the molecule at far of the sieve plate, it will move very fast. But when they are already near the sieve plate, the molecule will move at very slow speed. And then this slow speed will force the molecule at the upper part of sieve tube element, forced out from the upper part to lower part by active transport. So molecule will join into the next cycle again. So normally different solute move through the sieve pore at different rate due to the different molecular characteristic. So both energy streaming and transfer the sieve plate would be energy dependent, explaining high turnover of ATP in film cell. And then this process is require high energy or ATP lah, compare high energy or ATP because it actively transport process. And final one is about peristalsis wave. So it is one type of wave that we produce in the plant that transport the solute particle from the source to the sink. So normally this one is that it is using the cytoplasmic filament that found in the phloem sieve tube. So in previously we used about the phloem sac is transported inside the sieve tube anyway. But for this one peristaltic wave, the sieve tube element, the phloem sac actually is transported between the cytoplasmic filament. Okay, so between this cytoplasmic filament, there will be contract and relax. The cytoplasmic filament can be constrict and relax one, like muscle, like human muscle one. So this one will push the phloem sac from one sieve tube to another sieve tube. And then this process is also require energy because it is in the opposite direction depending on the metabolic energy and also ATP lah. So it is using the human muscle contraction and relaxation. It contract and relax, like here it contract and push the phloem sac from upper part to the lower part, contract relax until it reach the sink lah. So this one, when it is contract and relax, it will produce the peristaltic wave. So this is the theory about the peristaltic wave hypothesis. Okay, so actually today class is already end. Yeah, today class is end. So I already finished up the translocation topic. So today we just learned about the translocation based on the 4 hypotheses: bachelor flow hypothesis, electroosmosis hypothesis, cytoplasmic streaming hypothesis, and also the peristaltic wave hypothesis. Okay, so I think that's all for today. So any questions so far? Okay, so if no question, then we will end the class today. So we will see you again next week. So next week I will enter chapter 9 control and regulation. So thank you. So thank you for attending and see you next week. Bye.